Aging is shaped by genetic instructions, environmental exposures, and a wide range of social, emotional, and behavioral factors. Cognition and mental health sit right at the center of this process, influenced by everything from sleep patterns to stress to what our cells are doing when no one is looking. Neuroimaging has given us a front row seat to how the brain changes over time. Measures like cortical thickness reveal subtle thinning in specific regions, while the hippocampus and other subcortical structures show volume changes linked to memory and learning. Together, these features map the brain cognition pathway, showing how structure supports function and how that support can weaken during pathological aging. Different regions age differently. The medial temporal lobe, essential for episodic memory, is often in the early site of change. The parietal lobe helps integrate sensory information and supports attention, while the frontal cortex manages executive function, planning, and self-control. Posterior pathways sometimes show early disruption, and the brain may attempt short-circuit or compensatory pathways to maintain performance. These work for a while, but they are not infinite. When compensation starts to fail, mild cognitive impairment can emerge. Risk factors include the ApoE, omega-4, alterations in folate levels, and metabolic markers such as triglycerides and lipoprotein cholesterol. These biological signals don't determine destiny, but they do shift the odds. Mood and cognition are tightly intertwined. Somatic depressive symptoms often correlate with changes in the posterior cingulate cortex, a key hub in the default mode network. This network, linking the medial prefrontal cortex, parietal cortex, and hippocampus, supports self-reflection, episodic memory, and working memory. Functional connectivity within this network can weaken with age or disease, altering how thoughts flow when the mind is at rest. At the cellular level, aging brains show changes in neurogenesis, especially in the hippocampus. Astrocytes and microglia, once quiet support cells, can become chronically activated. Glucose handling becomes less efficient. Neurons struggle to maintain energy balance, and chronic inflammation settles in as an unwelcome long-term guest. Progressive cognitive impairment reflects more than memory loss. Affect motivation and emotional regulation change as neuronal dysfunction accumulates. Many psychopathologies share overlapping neurobiological causes. The monoamine theory highlights the role of biogenic amines, serotonin, norepinephrine, and dopamine in mood and cognition. These systems don't operate in isolation. They interact with stress responses, circadian rhythms, and metabolic states. Aging then is not simply time passing. It's biology negotiating change one pathway, one network, and one memory at a time.